maybe we can now go back to our slideshow. Where's that, Gordon? No, down here, is it? Yeah. Okay. Pathotype TTKSK is not alone. And this is where things get out of date a little bit. This was put together about 3 years ago, and there are now how many, Dave, Zach?
Officially 8, but unofficially more.
Okay, so 11. Yeah. So how many have I got here? 1, 2, 3, 4, 5, 6, 7. These are all pathotypes that have been identified in the ug99 lineage. Here's the original isolate here, TTKSK. These are all variants that we believe have arisen through mutation, simple mutation in that lineage of the pathogen. I've got only a few here. Since then there have been another 4. Is that right? Yeah. So there are another 4 pathotypes that should be illustrated on this diagram that have also are related directly or indirectly to this first pathotype that was identified, this TTKSK. And you can see here again these countries would be, I think, also out of date quite a bit. Yep. One of Dave's diagrams here just showing, and again 7 mutants, there are now 11, but showing a different coloured dot. This is, as I said, one of Dave's diagrams for each pathotype, right? So this yellow one here is the, where are we, TTKS, red. This is the original one here, TTKSK. You can see that at the time this diagram was put together, it had been recorded in Uganda, Tanzania, Kenya, Ethiopia, Sudan, Yemen, and up in Iran. That's the original TTKSK pathotype. All right? So, Dave, I'm sure you're going to give us an update on that, and Zach, again, will run us through the North American system. But the important thing to realise is that ug99 is not the pathotype name, it's the sample, the name that was given to the original sample that was collected in Uganda that contained the pathotype TTKSK. All right, and the other significance of this is that there's more than one pathotype in the lineage. Sometimes I go and speak to farmers in Australia and they say, what are we going to do when ug99 gets here? And I say, which one? And they say, what do you mean? And I say, well, there's actually 7 of them. And really, because we know what resistance genes we've got in our varieties, to be really honest about it, the thing we're really worried about is this. We're not so worried about, well, we're worried about this, but what we're really worried about is this, because SR24 is present in a lot of our varieties and it's effective. So virulence on SR24 would be far more dangerous to us than virulence on SR31. Okay, so the threat is to us as these exotic incursions are occurring, pretty big from any of these, but this is the one we would probably be most worried about, this one down here. Okay, I think some of you would have some familiarity with DNA fingerprinting, right? So we can actually do DNA fingerprinting using sequence information or markers like microsatellites. And the DNA fingerprinting helps us to resolve, let's say, genetic relatedness of rust isolates. So this is what we call a dendrogram, a tree, showing 61 isolates of the wheat stem rust pathogen, Buccinia graminis triticide, that have been sequenced. This work was done by Dr. Les Szabo in North America. And the dendrogram is based on 3 million markers. That's a lot. 3 million polymorphic, what we call SNP markers. I won't go into the detail now, but that's like looking at 3 million different traits in the pathogen genome and comparing them across those 61 isolates and then drawing a figure that shows things, shows the similarity between things. So things that are far apart on the tree, like this one down here and this one down here, we would say are genetically very, very different. Things that cluster together, like these 2 from Australia here, we would say are genetically very similar. And the thing to notice about this is that all of these isolates here are in the ug99 lineage, right? They're all isolates of pathotypes within that lineage. And this is showing us that they are genetically discrete. So this is a genetic lineage within the wheat stem rust pathogen population that seems to be very distinct, and they all seem to be very similar. So this is supporting the idea that these things have arisen through mutation in a common genetic background. All right? One of the really big surprises here was that when Les did this work, he went back into his historic rust collection. He has a collection of many, many stem rust isolates that he kept in liquid nitrogen, and he pulled out one from Ethiopia from 1972, and that falls in this ug99 lineage. So that suggests that the genotype within which the ug99 family sits has probably been around since 1972, and possibly even longer. All right? So what's probably happened is that there's been a stem rust genotype out there, and over the past 30 to 40 years it's just acquired the virulences that have made it important through those single-step mutations that I showed you with the stripe rust before. Okay, so it's not ug99, does not appear to be something, this TTKSK doesn't appear to be something that's just jumped off Barbary with all that package of virulence genes in place, in place with the ability to attack all those varieties. It's probably accumulated those virulences over a period of time.
Robert, in the wheat atlas, the rust resistance gene, I think there's a report in there from Julio about virulence
There is.
on SR31.
Yeah.
Yeah.
Confirmed?
But it does say it is not confirmed. It
Yeah.
does say in the manual.
Yeah, so I don't know if, and again, you're going to get you've got the PDF of the book.
Yeah.
Yeah, so I, together with two of my colleagues, 20 years ago now, back in 1995, we published a book called Wheat Rusts, an Atlas of Resistance Genes, where we describe each of the rust resistance genes, their chromosomal location, their use in agriculture, and their effectiveness. And in there we referred to Julio Huerta's PhD thesis. I don't know if any of you know Julio here, some people do. Julio works with Ravi and Simmet. He did his PhD in Minnesota quite some time ago, and he looked at a global collection of stem rust isolates and a global collection of leaf rust isolates. And in his studies he actually found what he believes is a single isolate of stem rust that was actually virulent on SR31. So this is a long time before we found ug99, but it was never confirmed, I don't think. And unfortunately, I don't think they can find that isolate in their collection. So, but anyway, that it was referred to in our book, and I think from memory, I think the sample came from Turkey, possibly, that Julio thought had virulence for SR30. I could be wrong on that, but anyway.